Consider this before viewing Human Genome, Part 1. What do you know about DNA and its role in building unique genetic structures? As you watch the program, consider the benefits of knowing the sequence of genes that make up human DNA. Consider how an understanding of the human genetic code may influence the future of our species. Assignment Discovery now presents Human Genome, Part 1.
The human form is the remarkable product of millions of years of evolution. Only in the last blink of an eye have we started to understand the wonders that have shaped us. We now know that the graceful human machine is in many ways a product of the genes within us. Over the past decade, scientists and labs around the world have mounted an all-out effort to read our entire genetic blueprint. Efforts in private industry and in government-sponsored labs have paid off handsomely, as researchers have almost completed decoding the entire human genome. In the United States, Francis Collins heads the federally funded part of that effort, the Human Genome Project.
The human genome is basically all of our DNA, the complete instruction book. Every organism has a genome. Bacteria do, and roundworms do, and flies and yeast and dogs and cats. Ours happens to be 3 billion letters long. It's arranged onto chromosomes, but when you're talking about all of it, you're talking about the genome.
The Human Genome Project is the attempt to define the complete genetic code, the 3 billion letters that contain all the information about human heredity. From the keratin in your hair, to the hemoglobin in your blood, to the smell receptors in your nose. All that's written in the human genome, and we want to read it out so we know the parts list.
Human beings are more than simply the sum of all those parts, but those parts make up the basic building blocks of our bodies. We are made up of 100 trillion cells. Inside nearly every one of those cells lies a natural memory bank, more compact and efficient than the best computer chip. The living human memory chip is composed of 23 pairs of chromosomes, microscopic threads of life. That's where our genetic code lies. Each chromosome is made up of a single, seemingly endless strand of a material called DNA. If the DNA in one single cell were unraveled and laid out straight, it would stretch for nearly 6 feet. Most of that DNA doesn't seem to do anything at all, but about 5% of it carries coded instructions. As scientists read that code, they expect to find about 50,000 words. Each of those words is a specific instruction for building one small part of us. Each instruction is called a gene, shown here in purple. Nearly 50 years ago, James Watson and Francis Crick discovered how these simple threads of DNA orchestrate the secret of life. They found that DNA has a hidden structure that makes it ideal for carrying information and also ideal for passing it along from one generation to the next. A DNA molecule can be unzipped down the middle, and from each half, a perfect copy can be formed. That's how the information on DNA passes along when cells divide, and how the information is moved from one generation to the next.
DNA is this fundamentally interesting and important molecule. It is, if you want to talk about the center of life, it's got to be this molecule, because it does, after all, play the tune that the rest of biology dances to. If you want to think about it in musical terms, you could even say, well, DNA is the score, and we are reading the musical score for human beings, and that's pretty heady stuff.
A DNA molecule is shaped like a twisted ladder, a double helix. The legs of the ladder aren't remarkable, but the rungs are. That's where the tune of life is written. At first, that melody doesn't appear to be very interesting. It's composed simply of 4 notes, A, T, G, and C. Those letters stand for DNA building blocks called bases. These bases pair with one another to form the rungs of the DNA ladder. A always bonds with T, C always bonds with G. Our entire genetic code is written in this simple language. It's like an encyclopedia written with just 4 letters, and it's 3 billion letters long. To gather all this information into our cells, the DNA is wrapped around proteins, bundled up, and then coiled up into 23 pairs of chromosomes. Our genetic information lies on those chromosomes. The chromosomes are the volumes that make up the book of life. These books don't have pages. Each volume contains a continuous stream of information. The machinery inside our cells can read the DNA code. Cells make use of that information to carry out their jobs, and they also pass it along to future generations. Scientists are now completing the task of reading those volumes cover to cover. Much of that work has taken place at the Whitehead Institute in Cambridge, Massachusetts. Here, scientists have been breaking chromosomes down into tiny pieces. Then they read the DNA that's contained on each of those pieces to identify the exact sequence of the A's, T's, C's, and G's. That's called sequencing.
Sequencing a genome is in a certain sense like reading a book, except that you can only read it one sentence at a time and you don't know where the sentences come from. So it's like taking a book, shredding it up into lots of copies, scattering it on the floor, reading the sentences, and patching it back together. The current tools for biochemistry only let us read short stretches of 500 or 700 letters at a time. And so the whole trick of sequencing is to take a human genome, tear it up into pieces, read the pieces, and reassemble it.
The DNA from this enterprise comes from blood samples donated by a handful of anonymous volunteers.
Everybody is 99.9% identical at the DNA level. So it doesn't really matter who we pick, because we're all the same but for 1 letter in 1,000. So when we talk about the human genome, yeah, it's true we're all a little bit different, but we could sequence anybody in this world and largely entirely get the same basic information about the parts list. It's possible now to generate millions and millions of letters of sequence per day. In the course of an hour's television program, 600,000 new letters of DNA have appeared from the national databases. Access to the sequence is no longer the rate-limiting step for progress. We have more sequence in a way than we fully know what to do with, and researchers are instead focusing on developing new and better methods to do things with that sequence.
Our DNA sequence is mostly the same, one to the other. And by studying the 99.9% of it that we all share, we learn a lot about the basic processes of human biology, things about development and how you go from being a single-celled embryo to being this incredibly complicated organism with 100 trillion cells and all sorts of different tissues and properties. That's pretty amazing. But by studying the part where we differ, we also are learning why it is that some people are at risk for some diseases and others for other disorders. And very soon, certainly in a matter of 10 years, we should be able to offer people the chance to find out what their risks are. And perhaps another 10 years after that, a whole generation of new therapies for disease will arise out of this information.
Already, some of those stories have been revealed. Limone sul Garda is a small village in the Italian Alps. The people of this town look like ordinary folk, But a few lucky people here harbor a secret in their genes. This trait, passed from one generation to the next, lifts their worries about heart disease. Felicita Fava is one of the lucky few. She's 76 years old and eats whatever she wants. A lifetime of consuming one local delicacy, oil-marinated pork sausages, could spell trouble for many people. But not for Felicita. Her doctor checks her blood vessels using ultrasound. Hers are remarkably clear. Many women of her age have vessels that are clogged up with cholesterol, reducing blood flow to the brain and increasing the risk of heart disease and stroke. Valerio Dagnoli's family moved to Milan, 40 miles from Limone. He also carries this protective trait. Scientists interested in getting to the root of this remarkable story have been studying his family. Doctors checked cholesterol in Valerio's daughter Valeria and found that she inherited this protective trait, so she doesn't have to think twice about cholesterol. Neither does her brother Marco. In this fortunate family, another generation is on the way. Marco and his wife Paola are expecting their first child. Scientists came to realize that their healthy blood vessels result from a simple inherited trait, and anyone who carries that trait has a 50-50 chance of passing it along to the next generation. Thanks to some remarkable scientific detective work, doctors tracked down the precise genetic code, the gene that's responsible for this trait. They call it A1 Milano. Its secret lies inside Marco's cells, somewhere on his 23 chromosomes. But where? By studying the Dagnoli family and others, researchers tracked down a bit of genetic code that's passed down to people who inherit this rare trait. That DNA sequence lies on chromosome 11, and it turns out that only one letter has changed, but that is enough. To understand how genes work, it helps to know a bit about how the human machine is put together. The cells that make our muscles, organs, and other tissues are made up of proteins, fats, and sugars, along with a pinch of DNA. Proteins are the most important parts of us. They are the molecular machines within us. DNA code tells the body how to make proteins. This particular genetic instruction tells the body how to build a protein that circulates in the blood. Proteins are made of a string of building blocks called amino acids. There are 20 amino acids, which make up our proteins by being combined in different orders. The order of those amino acids is dictated by the code written in our DNA. Each amino acid is written with a 3-letter code. In Marco Dagnoli, a single letter of the genetic code has been changed. As a result, the 3-letter code CGC, which specifies one amino acid, becomes the code TGC, which specifies another. One little change, but what a difference it makes. That difference isn't obvious until you penetrate deep within a cell. The cell's inner sanctum, the nucleus, houses our chromosomes. Here, a molecular factory works to tap into the genetic information. Specialized enzymes unravel the DNA, and this machinery splits the DNA ladder in two and reads the code. Enzymes make a mirror image copy of the DNA by matching up the A's, T's, C's, and G's with their appropriate partners. The new copy is called messenger RNA. It carries the code for a single gene. Once an entire gene has been read, the strand of messenger RNA is complete. It moves out of the nucleus into the main compartment of the cell. There, a different set of enzymes takes over. Their job is to translate the genetic code. They read the genetic code 3 letters at a time. Each 3-letter word spells out the code for one amino acid. Other enzymes fetch the amino acid that's being called for and tack it to the end of the growing protein. Amino acids are added one at a time, like pearls on a string, as a protein gradually takes shape. The amino acids determine how that protein folds up, and that folded shape, in turn, determines how the protein will work. Change just one amino acid, and you can change the shape of the whole protein, for better or worse. Luckily for Marco and his family, when their body reads this code, it makes a protein that turns out to be very adept at clearing cholesterol out of the bloodstream. That protein is just one out of the tens of thousands that make us who we are. Individually, they seem insignificant, but altogether they create the mystery and the beauty of the human body. Some genes tell our cells how to make the proteins in our muscles. They give us the freedom to move. Others govern the building of our skin. Still others can react to light. Those are the proteins in our eyes that give us the gift of sight. Most of the time, our genes carry faithful instructions to build all the proteins we need for life and health. But when something goes wrong in a gene, that can make us sick.
There are probably about 2,000 or 3,000 genes where a misspelling in that gene is known to cause a particular disorder. Most of those are pretty rare conditions that may only affect 1 in 20,000 or 1 in 100,000 people. More recently, though, the tools of genetics are allowing us to look at conditions that are inherited in much sort of less obvious ways, things like diabetes or heart disease. That's where many people see the most impressive payoff from genetic research coming, because we're talking about millions and millions of people who are at risk or affected by those disorders. Only a few of those have been unraveled so far. But we're going to see in the next very short number of years that frontier being blown wide open by the ability to use this instruction book that's getting developed right now and information about misspellings and variants in that book to draw these associations and unravel those mysteries.
Biologist Alan Wilson realized that he could use DNA samples taken from people all around the world to figure out how we are all related and where we all came from. Wilson focused on a special little piece of DNA, the one little bit that is not part of our chromosomes. This DNA is found inside small vessels that float inside our cells. These vessels are called mitochondria. Due to a quirk of evolution, they carry their own DNA. It is a lot like the DNA on chromosomes, only it forms a loop rather than a strand. And we inherit mitochondria only from our mothers, so they trace a unique history of our maternal ancestors. Slowly over time, the DNA in mitochondria develops small glitches, mutations, not enough to harm them, but enough to be noticeable. Knowing these simple facts, Alan Wilson was able to make a huge leap to understand where human beings came from and how they migrated across the globe.
Human race is essentially one big family. descended from a group of maybe 10,000, maybe even fewer than that, folks who lived in Africa about 100,000 years ago. That's a very short period of time, maybe only 5,000 generations separating all of us on this planet from this common pool of ancestors. Genetics has taught us that. It's also taught us that most of the variation in humanity pre-existed in that original ancestral pool. And so most of the variability amongst us is variability that you find in every ethnic group, in every population. And the differences between ethnic groups and populations are actually only a very minor component of human variation in general.
In the future, we're not going to evolve randomly. We're going to evolve in a directed manner. We're going to take control over our own evolution. And the reason it can happen so much more quickly is that we're going to decide generation after generation, or people will decide who have access to this technology, what evolution will be.
Lee Silver foresees the day when a simple computer program can interpret the DNA in human embryos and easily pick out flaws in them. It could scan for some of the serious genetic diseases. A computer program could also look for genes that simply increase the odds of developing a common ailment, such as cancer or heart disease. Scientists could also scan for traits that are more likely to be protective.
Parents will be able to choose which combination of genes they would like to see in their child. I really believe that the point will be reached when they'll actually be able to see a picture on a computer screen of what the child would look like if this embryo is allowed to become a child.
It might be tempting if it were possible to give your child a gene like the one that protects the families in Limone from heart disease, but would it really work? Francis Collins has his doubts.
How would you actually go in and make that change without being absolutely sure that something hadn't gone wrong and you'd actually changed a few other places in the genome at the same moment without meaning to? We have no technologies that are that kind of surgical correction of a single letter in the DNA code. We're not close to that. And even if you could do that, what's to say that we won't discover in some future environmental circumstance that change is actually bad for you in some environments. Maybe it helps out if you happen to live in Italy at the present time, eating the diet that they're eating. But maybe in some other circumstance, that variant would turn out to predispose you to Alzheimer's disease or diabetes. We don't know that.
Marco and his wife Paola are simply letting Mother Nature take her course. It's a healthy boy, and yes, he carries the protective trait that makes this family notable. And with this child is born a reminder: we don't need to manipulate our book of life to appreciate the awesome power of the gene.
Now that you've seen Human Genome Part 1, talk about this. In 1990, the Human Genome Project began identifying all the genes that make up human DNA. What are some of the challenges of deciphering the human genome? Debate the moral and social implications of this project. Now try this. Create a timeline for the scientific research done on DNA during the 20th century. Begin with Watson and Crick and conclude with the work on the Human Genome Project. Log on to discoveryschool.com for curriculum materials and resources to support Human Genome. To learn more, Assignment Discovery suggests Genetic Engineering by Clarice Swisher. Consider this before viewing Human Genome Part 2. Scientists have proved that some types of cancer are related to an individual's genetic code. As you watch the program, look for technologies that allow us to alter our genes. How might these methods help eliminate some diseases or increase human lifespan? Assignment Discovery now presents Human Genome, Part 2.
I think the most exciting aspect of human genome research is its potential to cure illnesses that we currently don't have good answers for. And I was frustrated by so many conditions that we don't understand at nearly enough detail to be able to design a therapy that's going to work. And so we run around treating secondary effects and putting our fingers in the dike and hoping that something will pan out without really knowing what's going on. Genetics and the Genome Project offers the potential to open up this black box and shine a light inside and to be able to unravel what's really going on in diabetes or asthma or schizophrenia in ways that will change the practice of medicine profoundly, fundamentally, irreversibly.
Until recently, it's only been possible to study one gene at a time, but now new technologies are being developed to search for thousands of traits all at the same time, to identify genes involved in causing disease and genes that make us susceptible to ill health. Scientists are developing a new technology called DNA chips that can study thousands of genes at once. Here, tiny snippets of DNA are being anchored onto a glass slide so they can be scanned simultaneously. Soon you may be able to walk into a doctor's office and get a blood test that will scan for many diseases and genetic traits all at the same time. Researchers have been hunting down disease genes one by one for the past 2 decades. The systematic search for human genes will vastly accelerate the process of finding genes involved in all aspects of health.
We are going to learn about all of those associations of particular genes with particular illnesses. And we're going to learn that rather quickly. The major genetic contributions to the common illnesses of Western civilization, things like diabetes and heart disease and high blood pressure and cancer and Alzheimer's disease, Parkinson's disease, those are going to be uncovered in the next 5 to 7 years.
Those genes can be found deep within the cell. They reside inside the cell's inner sanctum, the nucleus, on long strands of DNA. These genes are scattered across our chromosomes. And as scientists explore our genes, they're finding that the common diseases have a strong genetic component. Eventually, they expect to find genes involved in every ailment.
We don't actually know the basic causes of most common diseases like diabetes or asthma or heart disease or hypertension. Today we treat symptoms. The point of the Genome Project is to have the tools to find the actual causes. The causes don't guarantee that we're going to be able to make therapies, but the knowledge sure beats ignorance. And so the long-term effect, I think, of the Human Genome Project will be rational therapies and in some cases cures for these diseases. That's pretty much the promise over the course of the next 50 years flowing out of the Genome Project. It's not going to happen immediately, but that sort of understanding is going to transform medicine.
Scientists have found dozens of genes related to cancer. The challenge now is to understand how damaged DNA, like this sample from a cancer cell, causes disease and death. Most cells in our bodies are supposed to grow and divide, but occasionally that growth will get out of hand. A cell will keep dividing endlessly. That runaway growth becomes a tumor. That's cancer. Cancer is one of the most urgent medical mysteries. 1 in 4 Americans will die as a result of malignancies. A cancer cell can also break off and get swept away through the bloodstream. If that malignant cell lodges somewhere else in the body, a new tumor can start growing. That's called a metastasis. These are the first pictures ever taken of a cancer cell slipping between healthy cells, as they do when cancer spreads throughout the body. But seeing cancer cells at work and understanding how they act are two different things.
When we can understand the basis of a cancer, we can try all sorts of tricks for a therapy. It might be gene therapies, it might be small molecule therapies. The key is having an understanding of what's wrong. We've been trying to treat diseases without even knowing, in many cases, what's wrong. And it's a triumph that we've succeeded in many cases, but it's dependent on luck. The idea of having the complete inventory of the parts and where they work in the body, in health and in disease, means that we don't have to depend so much on luck. We can be systematic, and that's a real engine of progress.
The typical American has a 1 in 3 chance of developing cancer. But some people, like Lindahl Southern, inherit a genetic trait that makes cancer almost certain. Lindahl's mother developed breast cancer at the age of 28 and died 4 years later.
I had cancer at the ages of 18 months, 14, and then 30 and 34, I guess. I don't even remember my own. Isn't that sad?
You can always put your ear back in.
Lindahl's battle against cancer of the jaw left scars on her face. She decided to become a nurse so she could help other people who are living with cancer. Her genetic condition is rare, but it turns out that her disease involves a gene that is central to the story of most cancers. Lindahl's story involves a vital protein that's present in all our cells. It's called p53. These proteins are nursemaids of a sort for DNA. They bind to our DNA and stop our cells from dividing at the wrong time, so they can stop cells from growing out of control and becoming cancerous. P53 proteins work in concert with other proteins to help maintain cells in a healthy state. Cancers can sometimes start when another gene sends out an erroneous signal, telling the cell to divide at the wrong time. P53 and its helpers can shut off this incorrect signal. In this example, a protein called RAS is malfunctioning. This is a common step in some cancers. RAS starts sending chemical signals through the cell into the chamber where the DNA resides, the nucleus. These molecules bind to DNA and tell the DNA to start making a copy of itself. That's the first step a cell takes as it prepares to split in two. But if it is not time for the cell to divide, another protein will come along. Its job is to remove the chemical signal from the DNA, but it doesn't always win this battle. This is where P53 enters the picture. P53 binds to the DNA and activates some defensive enzymes. These defender enzymes then stop the inappropriate chemical signals. If p53 has done its job right, the cell won't divide at the wrong time, and runaway growth, cancer, is halted in its tracks. Lindahl has faced a lifetime of cancer because she was born with damaged p53 genes.
How are you? I'm glad to see you. You're great to come. Come in, come in.
As a result, she has a defect in her p53 protein.
How are you?
Everyone who inherits this malformed p53 is at high risk of cancer.
In your situation with mutations in
In about half of all cancers, doctors find that p53 doesn't do its job. That happens because the genetic blueprint that tells the body how to make p53 gets damaged. That's a common consequence of aging. In Lindahl's genetic code, just one pair of letters is missing. That flaw is in the instruction that tells her cells how to build p53. As a result, when her body builds those proteins, they don't come out right. These enzymes can't do their job. Her p53 protein tries to bind to the DNA, but it can't, and that makes Lindahl vulnerable to cancer. Lindahl has mustered a great deal of strength to fight her cancers and to raise her daughters. There is new hope for families who have inherited defective p53 genes, and there's also new hope for the millions of people who develop a mutation in this gene during their lifetime.
It's been discovered that if you take a normal p53 gene and put it back into a cancer cell that has a mutated p53 gene, that cancer cell will undergo apoptosis, or will kill itself, basically.
Researchers are trying to figure out how to transplant a healthy p53 gene back into people with cancer. This laboratory has genetically engineered a virus that causes the common cold, called an adenovirus. This one now carries a healthy p53 gene. The idea is to use this virus as a form of therapy, gene therapy. When this virus infects cancer cells in the laboratory, it delivers the p53 gene. And sure enough, once p53 gets into the cancer cells, the p53 stops the abnormal growth. The cancer cells die off. Curing cancer in a petri dish is much easier than curing it in the real world, but medical researchers are now trying.
So gene therapy refers to an approach to trying to treat disease where the gene is the drug. Basically, you're trying to deliver a gene to a particular place in the body and get it to work. And for a gene to work, that means it has to be used to make a protein. In the simplest situation, you're using it for somebody who has a defective gene that's not working, and you're trying to compensate for that.
Our bodies need oxygen to live, but oxygen can also damage our cells. That's called oxidation.
There is evidence that proteins and fatty materials and DNA can be damaged because of oxidation. So in a way, for a layman, you might say we're aging because we're rusting, like iron is rusting.
Every breath we take brings us life-giving oxygen. Blood rushes oxygen to all our cells, but oxygen can also be present in a harmful form. This supercharged oxygen is called a free radical. Free radicals are very unstable. They're likely to react violently with whatever they encounter. Some of these free radicals drift into the cell nucleus. That's the inner sanctum where our chromosomes are housed. When free radicals collide with DNA, they can damage our genes. They can alter the genetic code and create mutations. Living creatures have developed defenses against free radicals. Human defenses are quite elaborate. They include enzymes that are constantly patrolling our DNA, looking for mutations caused by free radicals. They do a pretty good job of repairing the mutations that free radicals cause. But it's a never-ending process, and sometimes the accumulated damage is simply too much. In that case, the DNA nursemaid protein, p53, takes command once again. p53 calls up enzymes that will destroy the cell. It's better to lose a damaged cell than risk that it runs out of control and becomes a cancer. Just as we grow old and die, so do our cells. So our tissues are constantly replenished with new cells. That's useful in places where cells are lost, such as the insides of our blood vessels. When old cells die, neighbor cells can divide to fill the gaps. But as we age, this process slows down, and it can eventually come to a halt. Cells can divide only so many times before they simply can't reproduce themselves anymore. This may be the ultimate timekeeper for human beings. A cell is in some ways like a candle. When there's nothing left to burn, the light is gone. This burning wick is not simply a metaphor. On the end of every chromosome is a bit of DNA. It's a simple sequence repeated over and over. It's called a telomere, and it plays a vital role protecting the end of each chromosome and keeping it healthy. Like the wick of a burning candle, telomeres get shorter over time. When a chromosome divides, it loses a bit of the telomere on the end. The enzymes that build DNA simply can't work on the very tip of a chromosome.
There's a gradual loss of these telomeres from the ends of the chromosomes. Each time the cell divides and creates a new set of chromosomes, a little bit more of these telomeres are lost each time. Eventually you get to a state where the telomeres no longer function to protect the DNA, and they gum up the cell, if you will. I mean, they stick together, they stick to the walls of the nucleus, and it becomes impossible for the cell to keep dividing.
When cells can no longer divide, the clock of life grinds to a halt. The next step for that cell is death. Here in Dallas, Texas, some scientists think that this process plays a central role in aging. So they are avidly studying the telomeres as Ponce de Leon searched for the fountain of youth.
We know that aging is genetic. We know that aging runs in families, that people who have parents that live into their 90s, generally they live into their 90s, and there are other families where individuals always die in their 50s or 60s. And so we believe that part of that might, in fact, be due to the telomere length that you were born with.
Jerry Shea wonders whether it might not be possible to rebuild telomeres, giving cells a new lease on life. He's studying an enzyme called telomerase, which can make short telomeres long again.
If you think about our telomeres as being the gas tank in an automobile, and that when we're born, we have a full tank of gasoline, and as we age, we use up the fuel, and when we're old, we're basically running on empty. And so telomerase is essentially like adding fuel to the tank to keep the car rolling.
The genetic blueprint for telomerase is on chromosome 5. This enzyme is able to slide along a strand of DNA and extend the length of telomeres. When telomerase is present, cells can keep on dividing indefinitely. Telomerase is only supposed to work in an embryo. Cells early in our life have to divide many times in order for a human being to grow from a single cell. Telomerase keeps that process going. What would happen if we were able to tap the power of telomerase to keep our cells growing and dividing indefinitely? Would that extend our lives? Would we become immortal? It's certainly not that simple, but the Fountain of Youth may not be an impossible quest.
It is probably going to be much more complicated than fixing a gene here and there if you're really going to talk about prolonging human lifespan to a significant extent. Not to say that in 30 or 40 years we won't see the arrival of some clinical trials trying to see whether the normal lifespan can be extended to some degree. But the notion that we will achieve biological immortality still seems very far out of reach. I think the death rate will continue to be 1 per person for quite a long time to come.
We may never see the day when the aging process is stopped, but even slowing that process to expand the human lifespan to 110 years or maybe 120 years has profound implications for a planet that's already struggling under the burden of its 6 billion human inhabitants.
If we ever unravel the molecular process of aging, we'll be faced with the question of, should we intervene in the process? Should we make small molecules that delay aging? Should we ever contemplate gene therapies that would produce people who age more slowly? I think these are deep and important questions. They're not fundamentally scientific questions. They're social questions, not to be answered by scientists alone, but by the society at large.
We will be able to manipulate more than aging as scientists of the 21st century gain power over the human genome. The technology that could help cure our most dread diseases could also be used to alter future generations. Scientists can tell us what's possible, but it's up to all of us to decide what's desirable as we harness the power of the gene.